I nearly ended the stream on you after like 2 seconds. Okay, well, here we are. I very nearly didn't make it today. I basically was in a mad dash to the airport today because they were closing flights down and didn't know if I was going to be able to fly back tomorrow. Turns out I can't fly tomorrow, but I might be able to possibly fly on Saturday. So, yeah, it's going to be a bit of a trip. I don't know. So watch this space. I might be here tomorrow if things are changing every 6 hours. So I'm basically not. I'm not too worried right now. I've got my cup of tea. Everything's going to be okay. Where is everyone? Good morning. It's a bit early for you. I've got my cup of tea. I'm settling in for my night's work. Right. Okay. Let me just close down some of these unnecessary tabs here. I don't think that was all that unnecessary, to be honest with you, that tab that I just closed. Got to find it again. Okay, so today we're going to be covering, again, I've tried to link a little bit of the first live class into the second live class, a bit of meshing there, so it's kind of like should drive home a few points if you stick around for both. If you're totally new, then basically the study at home exambulance free course from Taylor Tutors is going to give you some structure. We're going to do some extra lives on YouTube with you, so I'll just give you a very quick whistle stop tour where to find it. I think we're going to create a new page for it, totally for the exambulance, but yeah, we're going to be here, as I said, live with you, just going through finalizing the content for you AS students. I think we've got an announcement for you tomorrow, but check out the YouTube page a bit later on because we might post schedule an announcement for tomorrow, some updated news for you year 13 students. So let's go back to Zoom. I'll show you if you are interested in terms of getting into the exambulance, then I wonder if I can just do this whilst I'm here. Okay. It's got so many windows open. I'm not used to doing this without having two screens. It's totally the wrong website that I want to show you. That's where taylortutors.co.uk. That's where we're going. Now I can share my screen. I've got the right thing there finally. Okay. taylortutors.co.uk, you've got the big buttons here. It should take you into the course. You've got all the structure and bits and pieces there. All the recordings are going on YouTube in a playlist. So there's a year 12 biology playlist, year 13 biology playlist of all the past ones. We're going to put them in order for you. Okay, let's gather my senses again and get the chat visible. Let's move Zoom because I don't need to see Zoom right now. Okay. Today, a bit of biodiversity classification, maybe some sampling biodiversity, so like dropping quadrats, doing transects. We'll see how far we get. And again, I'm going to respond to you people who are here live. I am here to help you guys out, and we can go in the direction of whichever's going to help. I'm going to keep it generic to all exam boards, so nobody's going to be excluded. Obviously, in 45 minutes a day, I'm not going to be able to cover everything. If you do need any extra stuff, obviously all our materials there should you want it. So to start with, I'm gonna go to some questions at the end if you want some. I'm gonna do a little bit of like, oh, now I need to zoom back. I need a bigger monitor. Okay, let's share my iPad and jump into it. Okay. The species and taxonomy is what I titled this. I don't know whether I would title this to be the same if I were doing it. I'd definitely put some classification or some phylogeny in the title if I was being imaginative. So what is a species? My question to you, first of all. You'll see I always pretty much start with key terms. Key terms are called key for a reason. They are very important. So species. Those of you who've been using Taylor Tutors, do you like the template notes? Have you been using the template notes? How have they changed the way that you study? So they are organisms. You can say sometimes that they have similar characteristics, but organisms are the same species if they can breed or interbreed to produce fertile offspring. Oh, the template notes just keep everything nice and organized. You know, they give you all the structure obviously in my courses, everything's like laid out, color scheme. So you know how it's all going to go down. I find it, I'm not going to go into courtship behavior because this is an AQA only topic, but just if you are an AQA, purposes for courtship behavior would be recognize mates of the same species, to stimulate the release of gametes, to recognize members of the opposite sex, and it indicates that they are sexually mature, so that you're not a juvenile anymore, that you are able to reproduce. So it could be cricket song, stuff like that. I'm not going to put it on the page. It's AQA students only. So this is a template note from my AQA guide. Okay, what next? I'm going to actually stick in some key terms. So what do we talk about when we're talking of phylogenetics? So we've got classification. Maybe I'll just talk to you for a second here, actually. So we've got taxonomy. Taxonomy is putting things into groups. So categorization. And you call one of the categories, you call that a taxon. So that might be mammalia. Could be any sort of group. With these, it's a massive family tree. There's an organism we refer to as LUCA, the last universal common ancestor. So it was some ancient archaea, bacteria, single-celled, prokaryotic type thing, DNA genome that divided, and it was heritable, and it has spawned all of us. So if you literally like your small-scale family tree, that applies to all of us. All living things are cousins. We share a common ancestor to some extent. So taxonomy is looking at, and okay, so again, it's all a little bit academic because a little thought experiment for you, YouTube. Okay, so let's take a passport photo of yourself, face down on the table. This is in one of Richard Dawkins's books. And now imagine, if you're female, imagine your mother. If you're male, imagine your father. And you take a passport photo, headshot of them, face down on the table on top of yours. And then grandfather or grandmother. And you go down on the table. So you're just building a stack of photos face down with you at the bottom and your direct maternal or paternal lineages stacking up. You end up with a massive stack of these photos face down. And then you keep going on that stack. And at no point is any other photo any more different than you are to your mother or to your father. It's just very, I mean, they're not the same, but you're pretty similar. You go back 185 million of those photos, it'd be a fish. So at what point did you become a mammal? Or at what point? It's like, what point do you become an adult? Is it on the strike of midnight on your 18th birthday? So it's a continuum. So we divide things into species because it's convenient to categorize them. I'd love to just wax lyrical about speciation and biology and how in prokaryotes, again, not on the spec. I'm just gonna, if you want me just to talk to you about interesting stuff, then I can. It depends how keen you are to really get through this. So bacteria divide by splitting themselves, binary fission. They just make basically mitosis. They just make exact copies of themselves. And okay, so that means that you are all genetically identical to one another, which means if you become susceptible to a disease, to a pathogen, to an antibiotic, something like that, then pretty much all of you get wiped out. But we know bacteria are antibiotic resistant. How does this work? Well, bacteria can pass little loops of DNA. So those plasmids, they can basically put genes into those and pass them sideways. So not to their mother or to their offspring, but they can go to their brothers and sisters. So it's like sideways. They call it horizontal gene transmission. The process is called conjugation. If you're an AQA student, you're writing an essay, you could write some of these things down and blow your examiner's mind. But humans do it too, it turns out. It turns out that the early hominid species, there were probably myriad of various different hominin species. We know hominid, there's a whole bunch of human hominids that descended in, we thought East Africa until most recently in Morocco, I think it was, they found a species. So we got 50% older as a species. We thought we were 200,000 years old. We now know that we're at least 300,000 years old. And that fossil was found nowhere near where we were expecting to find it. We found it in North Africa, and we were expecting it to be East Africa. And, anyway, so when Homo sapiens made one of their waves out of Africa, Homo neanderthalis, the Neanderthals, were well colonized over the Eurasian plate. And we didn't, when I was at university, we were taught that you were not the same species. We didn't interbreed separate species, that we outcompeted them probably. Maybe we helped in their demise in other ways, but they just faded away. We didn't survive when we did. But we now know that we did interbreed, and 2% of most like European people have 1 to 2% of Neanderthal DNA in us. And so what we basically did is when we emerged out of Africa, we were pretty adapted to the relative warm. I mean, how much them being larger and having a smaller surface area to volume ratio being an adaptation to the cold, I think that's quite old school Neanderthal theory now in terms of what's up to date in terms of the research there. But what we did do is mate with them. And so sure, there's going to be a lot of weirdness going on, but they've been living there and adapted to those environments and picking up local adaptations by directional selection. They've been like picking up some of those gene mutations to be better adapted to their environment. So by mating with them, any of the really useful ones stick around. All the ones that screw you up and like cause deformations or abnormalities or things that are not a benefit, okay, you're going to die. It's the way of the world. That's natural selection. But those useful genes that end up in our genome as a result of interbreeding with what was a different species as we used to think, now that we were able to interbreed to produce fertile offspring, that is what introduces those useful genes into the population very quickly. There's probably 50,000, 100,000 years worth of adaptation by just having sex with the neighbors and just stealing some of their useful genes. Does that kind of make sense? And do you want me to do more kind of stuff like that, where I just tell you about biology and how awesome it is? Or do you want me to do like notes? Maybe I can do more stuff like that with the year 13s, and maybe you year 12s are a bit more focused on covering everything. I want to know, like, I can talk about biology all night, a bit of both. Okay, well, I'll intersperse some. Let's finish on that note. I'll just think of the next topic I mean, evolution is my bag. So when I did my university final year, it was like ecology, conservation, evolution. Human evolution was amazing, but the field, I don't know a single field that has been ripped up quite like human evolution since I finished university. Oh my God, I have to work hard to keep up to date in that field. Okay, so phylogenetics. That's where I started that rant from anyway. What do I need? Yeah, I need this. Phylogenetics is organizing things. So it's the taxonomy, but according in rank of evolutionary relatedness. So phylogenetic classification. To be honest, you can just say phylogenetics is organizing species into groups based upon evolutionary relatedness. Yeah, I'm going to say organizing, not just putting. Organizing species. Okay, so again, one of my students are gonna who follow the things that I say from time to time. So we have hippos split off at the Cretaceous Tertiary boundary about 66 million years ago in what's roughly today Pakistan, and they so their closest living ancestor. They're related. I think they're are they Perissodactyla? Are they odd-toed ungulates or even-toed ungulates? I don't remember. But so they are related to antelope. I think they're the odd-toed ungulates. Antelopes, pigs, camels. So their family tree, their phylogenetic family tree, would be linked to all of those like a regular family tree, except what's the closest living relation to the hippo? Some of you are going to know this because you're going to have heard me say this 100 times. Closest living, most recent divergence. Boom. Whales. Okay, so how about this for selection pressures? I'm going to go on another rant. I can make up this time if you're desperate to. I'll finish this page of notes for sure. Okay, so how is a pig and a hippo? They look relatively similar. Well, they have a similar niche, right? They're not that different. As soon as a whale has basically broke that relationship of having to return to land to give birth. So as soon as that cycle was broken, then there's a massive opportunity of all this energy in the ocean that they can go hoover up this energy that they didn't have access to. They couldn't get that far out. And because gravity is no longer a constraint, that basically opens the doors. So there's no penalty for getting bigger, other than you have obviously slightly more metabolism because you've got more cells to respire, and you've got more friction because of, but obviously there were probably other driving selection pressures in terms of predation. The bigger you are, like, it's probably more difficult to predate you. Maybe the bigger your calves are, and maybe calf predation is the pinch point in your evolutionary life cycle. So as soon as you break that relationship of having to return to land, then it's like boom, a rapid, like morphological or like your phenotype can change massively quickly because it's no longer constrained by gravity. And that's how such a massive morphological change can happen in such a relatively short time. 66 million years is not a lot for that. I mean, but when we've got strong selection pressures, when the environment changes that dramatically, you basically, like antibiotic resistance in a generation, all those that were not resistant are dead. The only survivors in the new post-apocalyptic antibiotic world are the ones that were resistant. So it doesn't take long. If there's a very strong push, it's like everything else dies. And then therefore only those that are able to survive do. It's just an extreme selection pressure. Okay. Go if I just let, it's kind of fun. I'm under pressure to just drive content home that or teach exam skills. It's kind of nice that I can just talk to you about stuff, give you an insight into like why biology is so awesome. AQA especially like you should define hierarchy. Anyone to define hierarchy in the chat for me? So it's basically a family tree. But let me just check I've spelled that right. And my spelling's not great. Do pull me up on my spelling if you feel like I've spelled something wrong. So it's, okay, groups within groups. I'm going to put, well, yeah, I do actually just put groups within groups with no overlap. Oh, dear. Between groups. This is basically only AQA are going to ask you to define that. It might be nice to know what it is. You're obviously most commonly going to see it in something that looks a bit like this. But you could also get a hierarchy drawn as, I'm not going to do my, like this. You might even get two species within a genera. So this would be like species, and then that would be genera. We're going to come down to that in a minute. Yeah, you don't need to know the definition of hierarchy. That is pretty much an AQA only thing that they'll ask you for something like that. Okay, so what is the classification system that we need? It starts with a D is the domain at the top. Domain was added relatively recently, 3 domains that was discovered through looking at the RNA in cells, basically. Who can give me the I'd recommend you make up your own acronym for this, because making up your own, the process of doing it, the thinking, that meshing together is what forms those neural pathways. It's so much more powerful as remembering something than someone just telling you it. I mean, if I tell it to you, it has to kind of stand out a little bit. So it is the domain kingdom, kindgom. Kingdom. Again, spelling. Is it 1 L or 2 L's in phylum? Phylum, yeah. Good. Phylum, class, order, family, and then genus and species. And my acronym that I use is dominant kinky people can often find good. Why not? We can have sex. Thanks to Alice Campbell for helping me come up with that. I think in fact Alice Campbell came up with that herself at university. Shout out to you if you're watching. Okay, so yeah, binomial naming system. Again, this is just basic facts you are expected to know. So we would be, what's our genus? So we have the genus with a capital, which would, and then we have the species lowercase. If you're writing it by hand, really it should be in italics. If you're writing it by hand, it's good biological practice to underline it. If you're writing it by hand, you will not be marked down for not doing that in the exam. We are, for example, Homo sapiens. In fact, some even say sapiens sapiens. I don't. So this is our genus, and this is our species, just in case you didn't know. Okay, and then phylogenetics. So basically, how do we know? How do we know whales or hippos are more closely related to whales than they are to pigs? How do we know that? Right, this is a relatively common question. I mean, in terms of what is the okay, bioinformatics. Can you give me any more detail on bioinformatics? So fossil evidence, similar and comparative anatomy. Yes, I mean, remember we asked you that question yesterday about what was not evidence for natural selection, identical fossils in the fossil record. Well, if it's identical, then it hasn't changed. It's not changing with time, and the definition of evolution is the changing frequency over time. So we can look at the genetics. Yeah, we can look at the DNA-based sequences, and then any of the other things that basically change as the DNA changes. So what DNA is going to give us the finest detail because it's got all the bases. What would be another molecule that we could compare to that would change as things mutate and selection pressures move that gene pool, shape that gene pool as to through differential reproductive success? Okay, so you could look at specific. So cytochrome C is a protein, a highly conserved proteins. Messenger RNA is going to code for those proteins, and the messenger RNA sequence. We can also look at the proteins, because the proteins will tell us what, if we can decode the primary structure, then we can obviously get some idea of which amino acid goes in which order, and therefore we'll know which triplets are going to code for them. But due to the degenerate nature of DNA, which means that there are more combinations of amino acids than there are amino acids, that we don't necessarily, we lose a bit of resolution when we go down to the amino acid level. We have much finer resolution of change logging. And here's a question for you. Are you more likely to see changes if you're looking for fine detail change between two closely related species? Would you look in the coding DNA or the non-coding DNA? Coding DNA being that which codes for proteins, non-coding DNA being either the introns within the gene or bits between the genes. I'd say the mutation rate would be constant everywhere. I don't think the mutation rate would change. Maybe I could be wrong with that, but I'd be pretty sure that the mutation rate is equal in the non-coding. Okay, so why in the non-coding? Because if it's in the coding DNA, it's functional. It does something. So if you make a protein that suddenly becomes non-functional, then you're at a disadvantage as an organism. It's more likely to be selected against for you to not survive and not reproduce. Whereas if it's in the non-coding DNA, if it doesn't do anything, I'm sure it probably has functions that we quite haven't got our heads around yet. But if it doesn't code for a protein, it changes, nothing changes. So you're going to just accumulate those changes over time, whereas changes that happen inside the functional core DNA that get translated into proteins that don't work as well, well, that's a negative. So then it's going to be more likely to be phased out of the population, just because exons are exported during well, from splicing, they are exported into the cytoplasm to be translated. So exons are exported, introns are got rid of. They are removed by splicing. Okay, so I should finish some stuff on phylogenetics here. It's only 25 past, we've got loads of time. So I don't know whether it's, let's just. So yeah, basically any of the molecules that are derived directly from the DNA. So we could say, here in my notes, I've written gene sequencing. I mean, that would be the best. You'd sequence base, 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 base. You just look at base by base. How many bases do you have in common? There used to be a thing in the spec about DNA hybridization where you just basically take two species of DNA, heat them to 95 degrees so that the hydrogen bonds between the parallel strands separate. You make everything single-stranded. You then stick some DNA from species 1 onto some DNA from species 2. It will anneal, it will stick to each other. And obviously, if it's really closely related, most of the bases are going to be much the same. And the bases, the A's are going to stick with the T's. It will stick together anyway, but like it will stick together better if the DNA is to a more closely related species. And then so what you need to do to like basically separate them again is heat it. And the melting point at which they separate is proportional to how many hydrogen bonds that they have. And therefore, the more closely related the species are, the higher the melting point of the DNA afterwards. But you don't need to know that for your spec. It's just interesting. Yeah, I'll do a quick summary. I have been a bit disjointed. Okay, so gene sequencing. So you basically compare the order of bases, compare the sequence of bases. On the genome, I'm going to say that. So that's your A, your T's, your C's, and your G's. Basically, the higher the percentage match, the more closely related. Humans and chimps. I wrote an essay at university. Humans are 98% chimp. Or was it 96? I think it's actually, in my notes, I've got 96%. So that's human and DNA bases in common. And I'm sure you can probably argue it a few percent either way, depending on which. The other one, I'm actually going to change my notes here. I'm just going to put, so this is basically DNA. We can also look at the messenger RNA for much the same reasons. Obviously, you might lose some because you're going to splice out those introns, so the messenger RNA might not have all the detail in. And again, the non-coding, the introns are going to accumulate mutations faster, so give you the more resolution. And I'm going to put proteins. Why if I put it in brackets? I don't need it in brackets. I'm going to put proteins. Typically, you use antibodies and use immunoproteins, basically, because okay, so we've got the primary structure. Means we can go, this is my shorthand here for amino acid. You can go to codon, and you can then go to DNA triplet. So you can sort of infer backwards. And the other thing you can do is you can look at the tertiary structure to see how well the final proteins react with one another, tertiary structure, by looking at basically antibodies, how well antibodies will stick to something, or how well antigens or antibodies by looking at complexes between We could do, what else we got here? Let's stop sharing, see what we've got going on in the chat. Okay, post-transcriptional is basically changes that happen to the expression of a protein, to the messenger RNA, after it's been transcribed. AQA students will need to know about microRNA, siRNA. There's other bits and pieces in there, but that's the year 13 stuff. Yeah, you could use DNA profiling. Yep. So you could definitely use DNA profiling. Maybe I can add that as a little, I'm not seeing my screen right now. I mean, there are lots of different ways. Generally, the questions here are gonna, if it's a question about DNA profiling, it will set you up with that. It would, I'm sure it would allow you to say you could use enzymes instead of antibodies as well. Yeah, any protein will give you some of that sort of history. The changes, the mutations that happen to the DNA are reflected in the proteins. I could put DNA profiling, which is DNA fingerprinting, if you're not sure what that means. Okay, I wonder if, what have we got in here? Okay, what is meant by a phylogenetic group? Are you gonna let me zoom in? Please let me zoom in, yeah. What is meant by a species? Okay, again, just like things just to note here, give me a pen back. You've got 2 marks for species. What is that then? Organisms, similar characteristics. If in doubt, you know, if you're expecting a 1 marker thing and it's 2 marks, then respect the marks. This is important. So you can throw in organisms with similar characteristics or that look the same, that are able to interbreed to produce fertile offspring. It's probably able to interbreed and it's probably fertile offspring. But I'd just be a bit with, you know, just chuck in that extra detail if you need to. What is a phylogenetic? Oh, interesting. So it's, could you say a taxon that is grouped by evolutionary relatedness? I'd say it's a group of organisms organized by or grouped by evolutionary relatedness. It's looking for, say, phylogenetic and then evolutionarily organized. If I were to answer that in as few words as possible, they would be my 2. I sometimes challenge my students to do 3 words per mark, and that would be an example where you're just trying to get to the nugget of what the mark scheme is saying. And when you get really, really, really practiced at these things, you can do things like that. So for this, I would do interbreed plus fertile offspring. Maybe it would be interesting for you to watch me do a paper like this. Just to model it. Possibly, if I was hedging my bet, similar characteristics. Something along those lines. Do you want to check? Do you want to see how my 3-word answers go? So this is when I was talking about seeing it as a, let's have the mark scheme in here somewhere. It's question 3 quite clearly. Okay, here we go. What have we got? Grouped according to evolutionary links, history, relationships, common ancestry, able to reproduce, to produce fertile offspring. Nailed it. And you can get this good. It doesn't take mega practice. Obviously, I've done a lot of practice, but it doesn't take, it's not voodoo dark arts. I can teach you this to you. Okay, this is super common. So, I mean, we've been through this. Oh, spell it properly, though. They won't mark you on your spelling, but it should at least be reusable. Class, order. Okay, and then obviously they've given us this information here. So we're going to need to apply that. We have to add 4 other taxonomic groups the tiger and the clouded leopard belong. Okay, so we've got which one's which actually. Okay, we've got the column headings. So tiger, we've got Animalia, Chordata, Mammalia, Carnivora, Felidae. Okay, so they're different. They're all the same up to here. Where have we got up to? We've got up to this level. So we've got groups within groups getting smaller, coming down to this level. So what's next? Well, now, let's get this in black. Now we've got two groups, not that here, all the way, we've got one group, one group, one group, one group. But here, we've got, why did that not turn to black? Make it join up. This can be Panthera. And this one can be Neophilis. No, Phylogeny, yeah. So these two are done. Tigris goes inside this one. And then we've got, the sizes make no difference. Nebulosa, that's a cool name. Well, if they wake up too late, I can't do anything about that right now. I'm in Australia. I'm coming back as soon as I can. Which are more useful? Okay, we've got classic family tree. We've got what is meant by hierarchy. That was an AQA question. We don't need to know any of that. Again, species. They like to ask you for a definition of species richness quite often. What is species richness? I'm actually going to, again, same question. I put it in here twice because it's very common. How are we doing for time? You have to calculate different indices of diversity if you are, so AQA and EdXL, it's the same. OCR do Simpson's index of diversity, but I probably don't have time to run through both. So, okay, so let's do, what is the difference between an index of diversity? This is the same for indices of diversity in general versus species richness. How are they different? Species richness is just the number of species in a population. You sample the population. There are 12 different species. You might have 1 kookaburra. You might have 10 kangaroos, and you have 1,000 wallabies. It doesn't matter how many of them there are. It's just you have a kangaroo, you have a wallaby, you have a kookaburra. So the species richness, total number of species, no information about the number of organisms. An index of diversity takes into account how many you might have. If you might have 5 species there, all equal proportions, exactly equal numbers of each, very, very even in terms of the species evenness. You might have 5 species there. You might have 1 kookaburra, 1 wallaby, 1 kangaroo, 1 sulfur-crested cockatoo, and 10 million flying foxes. And so you've still got 5 species, but there's 10 million of one, and there's actually 10 million and 4 organisms there. But the species richness would be 5 for both of those things. So the index of diversity, whichever one you calculate, takes into account, you need to know how many individuals of each species, as well as how many, obviously, species total. Species richness is just purely how many species are there. How rich are you? How much money do you have? How species rich are you? How many species do you have? Yeah, so the abundance is a very excellent word to use. Let's see if I can find something that there's one on standard deviation. Okay, so in fact, this is pretty classic. I'll just whiz through this one. I think is probably going to be a good example for all exam boards. Bit of scientists investigate the effect of different types of farming of animals on the diversity and the number of dung beetles. They determine the number of dung beetle species, and so the number of dung beetle species and the total number on intensive and their total number. Intensive, rough grazing, and organic farms. So we've got the type of farm, we've got the number of species, yeah, and we've got the total number of beetles. Okay. What is meant by species richness? Okay, so classic, what is meant by species richness? It's the number of unique species. In addition to the information provided, so other than the information you've got, what other measurement is required? What other measurement is required to calculate an index of diversity for dung beetles? That's an interesting one. I'll wait for you guys there. So what do we know? It looks like we know everything, right? But we don't know everything. We've got, this is total number of dung beetles. So on the intensive farm, we've got whatever, 3,000 just. But what don't we know? Well, here we've got on intensive, we've got 14 species. So of those 14 species, how many of each one? We need to know the abundance. We know that there's 14 species and we know there's 30,000 beetles total, but we don't know how many beetles of each species. So this is simple recall, like basically I just told you really clearly that that's what an index of diversity is, but we've got to be able to, what do we have here? What is missing? And that's the way the new spec's going to test you. Okay. What does explain, give reasons for what the standard deviations suggest about the difference in the mean total number of dung beetles between the different farms. So total number of dung beetles, we're using figure 2 is what we're doing. That's where we're coming. I might even just get rid of I want to get rid of that. Difference in the mean. Okay, so we've got, let's just look at this. I did my class on standard deviation not too long ago. What does it suggest here? Well, I could probably find a ruler somewhere here, maybe. Line. Red will do. So this is the smallest group. Oh, it's a giant line. Let's make that as small as I can get it. So that's our upper range from the standard deviation of the rough intensive or the rough grazing. That's, yes. Why won't you let me draw another line? That's our upper range there. Our lower range, sorry. Oh, I just want to draw another line. The upper range there. So we're pretty confident that the actual data points live within these realms, basically. So what does it tell us if those standard deviations don't overlap with one another? Come on, pen, you can do it. It tells us. It must tell us about the standard deviations somewhere. What do the standard deviations? I assume that we would get told that these are the standard deviations. It's probably the 2 standard deviations. I'm not going to go into those details right now. Okay, because there's no overlap, we can, when we're looking at our standard distribution, we can be pretty confident that within 2 standard deviations of the mean, that most of the data, basically this just means that when we're looking at our bell-shaped curve, that being the mean, if we come out, usually it's 2 standard deviations either side of the mean, but basically most of that data, there's only a little bit of the data here and only a little bit of the data here. That means if it falls within, maybe if I can zoom in and sort of draw my, again, let's make that a bit smaller. Having a great time trying to draw this. Basically, that means that's our data set. Or in fact, that's not quite right, because our data set's going to be like that, with the mean being there. And it's just saying that most of this data falls within here. There is a little bit of data that's higher than that and a little bit of data that's lower than that. But if most of that data falls, oh, that's a terrible normal distribution, falls within there, and most of this data falls within here, and these don't overlap with each other, we've got clear distance between here and here, then we can be pretty confident that the true value lives in this range somewhere. And the true value, we can trust it, that it kind of lives in here somewhere, because it's unlikely it's going to be in these little corners over here, because there's not very much of the data as we go to the extreme. So that is what those error bars mean. Does that help to visualize it like that? To like flip the standard deviation on its side and then sort of view the main body of this data lives within these two realms. And if they don't overlap, then it looks like these are two significantly different bodies of data. The means probably are different. Error bars are gone from the spec. Error bars are not a thing for the, I've obviously cut this paper. I'll probably like reduced some of the slightly less relevant bits from this question and squashed it. So I've probably lost a bit that tells me what those standard deviation bars are. They should tell you, they will, in your exam, you will get told these bars, because it could be range without anything to tell us. It could be the range. It's unlikely to be the range because they're nice and symmetrical about the high point on the graph about the mean, but it's possible. The last one I want to just look at before we pack it in and go over to, this is like an absolute classic. We've got intensive farms. Oh, I'm back at crazy thin pen. Intensive farms. Farmers have removed hedges to increase the land for grazing. This resulted in a decrease in the diversity of birds on these farms. Give reasons why removal of hedges causes a decrease in diversity, and specifically of birds here. 3 marks, got loads of space. Again, it's classic. Removal of deforestation here, removal of hedges there. Could be, maybe there were ponds and the ponds have dried up, but removal of a type of habitat. So habitat has been destroyed. You want to say that there's fewer what? It's a great word to throw in here. Less habitat. We're going to end up with fewer birds. Fewer niches is the word I would go with. So niches, there's fewer hedgerows, there's fewer nesting places, there's, you know, maybe less insects, there's so there's less food for them. For 3 marks, that's probably going to be about that. We probably don't need to talk about here. We want to sort of, is the species richness directly going to impact the plants? No, I would say that the answer here is going to be decrease in habitat, decrease in the number of niches, fewer insects, fewer food sources, therefore maybe fewer nesting sites, fewer bird species. The niche is defined as the role of an organism within a species. It's not a very good description of what it is, but if they ask me in an exam, that's typically how AQA will define it. I would say, oh, it's 10 to, I need to get going. Okay, let's just mark this question. So you can see the mark scheme for all of those. What I can do, how about this? Tell me whether you like this or not. It's going to be a bit, that's all my playlist from. Okay, so they're the videos in my course. If you are following along with the course in this PDF, obviously in my handouts, they will link to the videos. So you can just watch the videos from there. So you can see OCR do a bit more on this than everyone else. So if you are an OCR student, then we've done that question together. You're going to let me turn the page. So is that big enough for you to read? Pretty much. I'll make it just all fit on one page. Can you read that? I need a yes, and I'm just waiting for the delay right now. Wait for the delay, wait for the delay. Big enough to read or not? Okay, let's just do it like this. You guys can pause it. You can have some questions as well. Okay, the index of diversity question, the data is the same for all of you. Pause it there for the data, but obviously the equation is different depending on how you need to calculate it. So it's probably loads, and I'll whiz through the mark schemes. All the spreadsheets that I made for that's the actual question that you did. I think it was about as far as I got. I didn't do that one. Okay, sweet. Okay, any questions? Videos are locked. Yeah, I mean, like obviously you've got a subscription. All the courses videos are open. So if you want to like just study, I mean, there's heaps more. Obviously, if you want to really get into the depth of this, then the subscription's there. Error bars are not exactly. I actually can't even remember what error bars are. They're not in the spec, so I haven't touched upon them in ages. They're not exactly the same as standard. I don't know how error bars are calculated, to be honest. Don't think I've ever taught that. I don't think it was even in the old spec. They would used to put them on graphs, but you didn't have to know what exactly they were. Okay, I'm going to log off, take a break. I'll be back if you're at year 13 to do some stuff, a bit of variation. We'll do some Hardy Weinbergs. How do you get better at biology? One simple answer to that, tailored tutors courses, TT revision method, super easy. Ask the people in the chat. They'll give you a slightly less biased opinion. See you tomorrow, probably. Watch out for an announcement tomorrow from the TT channel. I may or may not be here to do my class, depending if I'm trying to get on them, if more madness happens. See you soon.